Okay, in this session we're going to cover the sorts of mutations that we see in cancer, look at specific mutational signatures that are characteristic of certain genetic defects, and look at the underlying cause of why cancers seem to have so many mutations which they shouldn't otherwise have. So in covering this we're going to look at spontaneous DNA damage and how that is repaired, because lots of DNA damage occurs just by spontaneous reaction with water. We're going to look at aberrant DNA damage caused by this group of enzymes called the APOBEC enzymes, and these are cytidine deaminases, so they can mutate DNA. We can then look at how DNA damage is repaired due to the DNA polymerases who accidentally put in DNA mutations and then sometimes correct them, and then sometimes other systems will come in and repair any damage that's left behind. And then we'll look at drugs that exploit defects in homologous recombination, and we'll look at exactly what homologous recombination is. So firstly we need to think about how a cancer cell can accumulate so many mutations in a single population of cells, because to go from a normal epithelium to a metastatic cancer we need dozens of mutations. Here the 6 characteristic ones are shown. And this really happens because of the mutator phenotype. This is the phenotype where cancer cells acquire mutations potentially at a 100 times higher rate than normal cells. So this allows initially mutations to occur by bad luck or inheritance. So in this classic model, what we call the Vogelstein model of colon cancer, these colon cancers were initiated by initially a loss of the APC tumour suppressor gene. That could be the first hit just by bad luck, carcinogens, that sort of thing. Or the first hit could be the inheritance of a defective copy of the APC tumour suppressor gene. Irrespective of what the first hit was, we've got one bad copy of the APC tumour suppressor gene. The second hit is then, again, bad luck, carcinogen, that type of thing. Which is now resulting in a cell which has lost both copies of the APC tumour suppressor gene. We then seem to get some sort of genomic instability. In case of mismatch repair positive cancers we will lose HMLH1 or HMSH6, so these are mismatch repair positive and then accumulate vast numbers of mutations. Alternatively, the other cancer cells could enhance APOBEC expression, and as we'll find out later, cause lots of C to T mutations, therefore creating genomic instability. We could also get chromosome instability at this point as well. So all of this genomic instability can lead to activation of oncogenes such as KRAS, which can then drive proliferation, and then other changes which lead the transition to full-blown carcinoma. So one of the more common forms of DNA damage is spontaneous DNA damage, which we call deamination, which is where cytosines get deaminated, so loss of an amine group, to uracil, as it can be seen here. And if this is not recognised by some sort of DNA mismatch repair system, then this can easily lead to a G to A mutation, which is effectively the same as a C to T mutation. So it's really important that these get recognised and repaired by some sort of DNA repair processes. Now, some enzymes, such as these cytidine deaminases and APOBEC, can also do this and are oncogenic. So looking at spontaneous deamination, here you can see all the 4 bases and what happens to them when they become deaminated. So adenine becomes hyperxanthine and guanine. Both of these create unnatural DNA bases, so these can easily be excised by specific enzymes that will recognise these, because this happens frequently. Cytosine becomes uracil. Uracil is a natural base of RNA, not DNA, so that should be recognised fairly easily as being in DNA, and therefore it should be excised, and I'll show you the pathway of how that is removed on the next slide. When it becomes more complicated, we've got 5-methylcytosine, which can be changed to thymine by deamination. The problem there is if this becomes this, if it's in double-stranded DNA, that's fine, because a 5-methylcytosine should be opposite a G, and next to it there should be a G, and opposite that should be a methylated 5-methylcytosine. So whenever you see a mismatch of a G to T, and there's a methyl group nearby on a 5-methylcytosine, you know that the T is in the wrong position. Now what I'll go through here is the base excision repair pathway, which deals with deaminations. So here we have a deaminated C, so this G is opposite a U. We all know that U shouldn't be in DNA, so an enzyme called uracil DNA glycosylase, you may come across this if you do quantitative real-time PCR. That enzyme is called uracil N-glycosylase. It's the same sort of enzyme. What that can do is cut out the uracil base, leaving a gap on the DNA here. We then get an enzyme called AP endonuclease, which basically means apyrimidinic, cuts the backbone and then allows DNA polymerase to fill in the gap. So we've gone from a G to U, and then we're going back to a G. See there. So back to this methylation problem. As mentioned earlier, some cytosines are naturally methylated, and these control DNA as an epigenetic mechanism. When these become deaminated, they form thymine. That is will create a mismatch, so you'll end up with a G opposite a T, which is obviously wrong. If this is not corrected and then those strands are copied, we end up with a G to A or C to T mutation. That's the end result, because this C has become a T. So this is the most likely base change to occur, because partly deamination causes C to T mutations, but also this change can occur due to the enzyme APOBEC. So this APOBEC enzyme can convert, it's a cytosine deaminase, so it can convert C to U or 5-methylcytosine to thymine, so that can affect both DNA and single-stranded DNA. Now the main role of this enzyme, there's different isoforms of the enzyme, but one of the main roles is an antiviral mechanism. So if you've got any single-stranded DNA or RNAs lurking around that look like they want to be mutated, to be deactivated, then this enzyme is involved in this process. So if you've got cells that have got chronic viral expression, it's thought that that can induce cytosine deaminases. These are also involved in mutating the immunoglobulin coding sequences. So if you remember that immunoglobulin genes are made up of exons which are spliced and cut back together to create antibody diversity, and in the antigen binding domains, individual base pairs are changed to increase variation in that region, and APOBEC related enzymes are involved in doing that. So that's a normal function. The problem is APOBEC enzymes can be overexpressed in cancers. So you can see from this study, say APOBEC, cytosine deaminase mutagenesis is widespread in human cancers, and we tend to see C to T or C to G mutations. In this study we see that APOBEC3A is overexpressed with long-term viral infection of HPV. So we find that HPV induces squamous cell carcinomas, and we find that the APOBEC enzyme seems to be able to drive increased mutation rate in these cancers, especially if you've got HPV hanging around for a long period of time. This enzyme becomes overexpressed, then you get these cancers with lots of C to T mutations within them. So since hydrolysis converts cytosines to uracils and 5-methylcytosines to thymine, and APOBEC can convert these in the same sort of way, we get a characteristic signature which is a CG pairing that becomes a TA pairing, whether it's a CG to TA or a 5-methylcytosine paired with G to become a TA. All of these processes drive this type of mutation. So when we look at cancers, we often see an overloading of C to T, so C to T, C to T mutations. Sometimes that can be due to APOBEC, sometimes it can be caused by something else, but we'll next look at some mutational signatures that point to one mechanism or the other. So this is what we call a mutational signature, and these have come from the Cosmic database. You can find them by following this link here. And if you base the nomenclature purely on mutating a pyrimidine, so a C or a T, to all of the possible bases, you can have C to A, C to G, C to T, TA, TC, TG. Now that's just on one strand, so A to C would be the opposite strand, but if we just keep it simple, you've just got 6 possibilities here, and that accounts for all possible mutations. For each possible mutation like this C to A, you can also look at the preceding base and the following base, so you can have ACA, ACC, ACG, and so on, all the way through all the different possibilities. So what we can do is look at the frequency of an ACA to become an AAA, so that's a C to A at this point here. And by whole genome sequencing of lots of tumours, we find that there are dozens of distinct signatures. We've got 30 distinct mutational signatures so far, some of which are diagnostic, some of which are unknown cause. So this one, signature 8, there's a weak bias for C to A's. For some reason, they're always where there's a C but not a G, so you don't get this where you've got C, G, but you do see it where it's CA and CC. So I've got no idea why this is, but there's this odd signature here that is probably characteristic for something, we just don't know what it is. In contrast, when we've got something like an APOBEC overexpression, we see a real over-representation of C to T mutations. And so here, the vast majority of the mutations in this particular cancer are C to T's with a few C to G's. So, and they're also mutations that are TCs, not CT. So if you've got TC something, this APOBEC enzyme seems to be able to mutate the C into a T. So they have very specific substrates in terms of what they can change. So this is a classic signature for one of the APOBECs. So this is the one that we just looked at, signature 2. There's also signature 13, which is also associated with APOBEC, where you can also get C to G mutations. I'm not going to why that is, that gets quite complicated, but you still see there are still quite a few C to T mutations, or you get C to G mutations and very little else. So both of these are characteristics for some defect in one of the APOBEC enzymes. Now, the presence of APOBEC overexpression, it can be easily seen by immunohistochemistry, or it can be seen by DNA sequencing. You pick up the signature. Now, this has an implication for the cancer, because if you've got a high mutation rate, it means that tumours accumulate mutations at a high rate. If you give a targeted therapy, you can be pretty sure the cancer is going to find a way around it. So if you give an EGFR inhibitor, you can be pretty sure that this tumour will pick up, I don't know, PI3 kinase mutation, RAS mutation, RAF mutation, and you'll get failure of that drug. So it's certainly a trend for high mutation burden cancers accumulating mutations at a higher rate, and therefore you end up with drug resistance. This is particularly obvious when you look at melanoma, which you've got a BRAF mutation. You give them vemurafenib, and these inevitably relapse from therapy very, very quickly. And melanomas are one of those cancers that express APOBEC very highly. They've got a ridiculously high C to T mutation rate, and what we find is those mutations, when we treat with vemurafenib and we get resistance, we get a C to T mutation in an oncogene downstream of BRAF, typically MEK or ERK, and they become spontaneously active, so therefore vemurafenib no longer works. On the plus side, these highly mutating tumours, because they've got such a high mutation burden, have got lots of tumour antigens. Tumour antigens are mutated proteins which elicit an immune response, so the high mutation rate leads to a better response to immune checkpoint inhibitors. Now, immune checkpoint inhibitors are things like PD-L1 and PD-1 antibodies. And what is happening here is the tumours have got a very high mutational burden, so they are being targeted by T cells. T cells can be switched off by PD-L1, so the tumours can overexpress PD-L1. Therefore, you can reawaken the T cells by giving an anti-PD-1 or anti-PD-L1 antibody, and that will effectively reawaken the T cells and allow them to attack the tumour. Now, the tumours that seem to overexpress highest levels of PD-L1 are ones that have a vast mutation rate and a high number of tumour antigens, and they may well be doing that as a survival mechanism to effectively keep them immune to T cell mediated attack. So high mutation rate, resistance to therapy; high mutation rate here, a more susceptibility to some sort of therapy. So we've seen that enzymes or water can create C to D mutations in DNA, but there are other ways in which we can get acquired enhanced mutation rate, and that is through DNA polymerase itself and through the inability of the proofreading activities and mismatch repair systems. So DNA polymerase makes mistakes, 1 in every 10 to the 5 bases. Then of these, 99 out of 100 are repaired by its own proofreading activity. So that means that every 10 million base pairs or so, a mistake will be put into the DNA that the DNA polymerase has not noticed. 99 out of 100 of those are picked up by the strand-directed mismatch repair system, which spots one base opposite another base that's incorrect. So it sees most of them and it lets the odd one through, so we end up with a generalized mutation rate of 1 in a billion. So you think of copying the diploid genome, you expect a normal cell to make 6 mistakes every cell cycle, which is an acceptable level of DNA damage. Any defect in this strand-directed mismatch repair can enhance the mutation rate by 100-fold. So the mismatch repair system has a number of enzymes. 2 of the main ones are the MUT-L homolog and the MUT-S homolog of the E. coli gene. So E. coli is a bacterium, MUT-S and MUT-L in E. coli are involved in E. coli's mismatch repair. In humans, it's a human MUT-L homolog 1 and MUT-S homolog 6. So that's where the names come from. These bind to the mismatch and scan the recently synthesized DNA for a nick in the DNA. This is a nick that's awaiting joining by DNA ligase. So here's binding of the mismatch, and then DNA around that region is scanned until we find a nick, and then we basically cut out all of the DNA from here to the mismatch, excise it, and allow DNA polymerase to fill in that newly synthesized strand and repair that damage. Now, the mismatch system has real problems dealing with repetitive sequences of DNA, and these are called microsatellites. This is where you get a sequence that goes TA, 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 and it is really difficult to copy without making a mistake. There are trinucleotide repeats, CAG, CAG, CAG. These are also very difficult to copy. A lot of these are non-functional. Some are functional, some are not, but the fact that they're difficult to copy allows us to identify tumours which are mismatch deficient, because these repeats, when they're copied, sometimes give forward slippage or backward slippage. So here we can see a piece of DNA that's being copied, and what you can find is that the strand that is being copied, you can form these little hairpin loops that effectively skips one of those CAG repeats. You can copy that and you're sort of missing out one of those repeats. Or you can have a situation where the newly synthesized strand forms a hairpin loop and effectively adds in. So you can either add in or remove one or more of those repeat units due to forward or backwards slippage. Now, this doesn't usually matter much because most of these TA repeats are in regions that are not encoding. Sometimes these CAG repeats are in coded genes, but adding one and taking one away doesn't really cause much of a problem, but we'll see later on where it does cause a problem. So the phenomenon of a tumour being unable to replicate these bits of DNA effectively is called microsatellite instability. So the microsatellites, as I mentioned, are these CA repeats, and within the normal population, individuals have got 2 alleles, one maternal, one paternal, and they might be different lengths, because within the normal population Historically, these have slipped forward and slipped back, so that people have just got different length alleles at different locations. So let's consider PCR amplifying one of these CA repeats. And what we find is that when we do this, individuals have got two different alleles at most of these CA repeats. So here you've got N is normal DNA, T is tumour DNA. So here you can see this individual has got an allele there, which is 2 base pairs bigger. The tumour is exactly the same. You would expect that. Here you've got a small allele, a large allele, and then the tumour's got exactly the same, a small allele and a large allele. So that's what you'd expect to see normally. In contrast, this patient here has got a small allele. So this PCR product is 138 base pairs. I know that because I did the PCR. And it's a tetranucleotide repeat. So if this allele shifts by 1 repeat unit, it will go up by 4 base pairs. So the normal DNA have got allele 138 and 160. The tumour DNA from that same patient have got allele 138 and 164, showing that we've got a single repeat allele slippage. Now diagnostically, this can be used to say whether a tumour is mismatch repair positive or not, and all we have to do is repeat this experiment on typically 5 repeat polymorphisms, and if we consistently see this thing happening in several repeat polymorphisms, then we can classify this as a mismatch repair positive tumour. So these TA and CA repeats don't really cause much trouble. They expand and they contract in the normal population. It's not a problem. Where it does matter is where these occur within genes. And I've been talking about dinucleotide and tri and tetranucleotide repeats. Well, another region that's very difficult to copy are mononucleotide repeats. So that's a run of Gs or a run of Cs or As or whatever. And 3 well-characterized tumour suppressor genes have got mononucleotide tracks within them. BAX, which is pro-apoptotic, has got this run of 8 Gs. The mismatch repair system often makes a mess of this, or DNA polymerase makes a mess of it, and if mismatch repair system is defective, that could easily result in the inclusion of an extra base or loss of a base, and that will cause a frameshift mutation inactivating BAX. Similarly, the TGF-beta receptor type 2, TGF-beta is antiproliferative. If you lose the receptor, you lose the ability to respond to it, and this has got a 10A repeat in there. So it's very common to see a 1 base pair loss or 1 base pair gain in the TGF-beta receptor, and that will inactivate that receptor. And MSH6 itself, which is a mismatch repair protein, the gene for that contains a mononucleotide tract as well. So that seems rather paradoxical, but that's just the way it is. MSH6 can be inactivated by a defective mismatch repair. So when we look at cancers that have got mismatch repair defects, we can find specific cosmic signatures. So in this case, signature 15, we get a characteristic peak of certain C to T mutations within there. We get other things going on which appears to have no specific pattern at the moment. But signature 15 is one of 4 different mutational signatures associated with DNA mismatch repair. In addition to these changes, we see, classic to what we've just talked about, a small number of short, typically 3 base pairs or less, insertions and deletions at mononucleotide repeats. So you tend to, so the cosmic mutational signatures don't actually show insertions and deletions, but signature 15 is characteristic for having these C to T mutations and these insertions and deletions. And for reasons that are not entirely understood, the other 3 mutational signatures with mismatch repair also have lots of C to Ts. They also have insertions and deletions at dinucleotide and mononucleotide repeats as well. There seems to be an over-representation of T to C mutations in mismatch repair defects, but very few T to As and T to Gs. So that's sort of, so what you can see is we don't know why you've got these, you just do. And we don't know why we don't get these, we just don't seem to see them. So finally, we're now going to cover homologous recombination, looking at how it occurs, how it's a DNA repair process, and how it can go wrong and how it can be exploited. So if we have a single strand DNA break, which is a nick in the DNA, it could be just a phosphodiester bond that's broken, or it could be something more substantial, but it's just a single strand break. If that is not repaired and a replication fork drives into a DNA nick, we will find that the replication fork breaks. This is a stalled replication fork, and it has to be somehow pieced back together. This is just DNA that is detached from this. So there seems to be no sensible way of linking these together. However, there is. This DNA can be copied from the newly made sister chromatid. So to allow resolution of this, what's going to happen is this piece of double-stranded DNA that's broken away from the replication fork, we're going to degrade this bit of DNA here, so that it's single-stranded, and then we can match this DNA onto this DNA because they are antisense to each other. And this is shown here. So there's the bit of DNA that we've removed this red bit of DNA here to create a 3' overhang, and then we can match this DNA to this DNA by this strand here effectively detaching from the bottom red strand and lifting up and matching onto here, so it's a perfect match, which has been shown here. So if we do that and then get breakage of the DNA here and here, what we can do is allow this replication fork to continue this way, and then we can start producing Okazaki fragments down here again. So that's what's happened here at this next step. This leading strand has restarted, having used a bit of what was newly synthesized DNA from the bottom strand incorporated up here. This leading strand has now restarted, and then subsequently the lagging strand with the Okazaki fragments can restart. So this is the restarting of a stalled replication fork. So homologous recombination, such as here, we've got newly formed sister chromatids, we've got breakage there. We can get error-free replication of this break by copying the DNA from here, so we end up with a perfect DNA copy. So homologous recombination is mostly error-free, no loss of nucleotides. If we don't have that sister chromatid to hand, so if this happens in G1 of the cell cycle, chromosomes are a single chromatid. There is no sister chromatid to go to as a reference. So we get non-homologous end joining, which is basically we get a double strand break there. All the cell can do is erode back these ends a little bit and then just randomly stick them back together. This is what we call a quick and dirty repair for non-homologous end joining. It makes a mistake, it takes out some bases. Homologous recombination is a clean repair, no loss of nucleotides usually. So we can exploit this in certain tumours which are defective in homologous recombination. And this works on this protein called PARP, poly-ADP ribose polymerase. And PARP is crucial for the repair of single-strand breaks. In the absence of PARP, a single-strand break will become a stalled replication fork. That can collapse, but then that can be repaired by homologous recombination. Now, PARP is normally present, but we're going to look at the situation where we can get rid of PARP and where this is going to be quite useful. So some tumours are defective for homologous recombination, especially BRCA1 and BRCA2 mutated tumours. We see these in a lot of breast cancers, especially hereditary breast cancers. So these are homologous recombination defective. So remember, homologous recombination uses the sister chromatid to restart a replication fork if it stalls, and then normally homologous recombination will take over, but in these cancers homologous recombination is not available. So if we take a situation where we've got a single strand DNA break, PARP usually acts to repair that, but in the absence of PARP, a single strand DNA break becomes a stalled replication fork which must be repaired by homologous recombination. In these cancers they can't do homologous recombination, so what we can do is block PARP with a small molecule inhibitor, and what we do is we force these cancers to go down non-homologous end joining, which is error prone. So if you can create some single-strand DNA breaks, treat them with a PARP inhibitor, that will cause stalled replication forks. These cancers can't undergo homologous recombination, and they will undergo non-homologous end joining, incorporate loads of mutations into the DNA that is incompatible with life, and this will cause catastrophic DNA damage and death, and we call this synthetic lethality. So this is shown here. We've got functioning PARP enzyme, a single strand break. PARP will help to convert that and create a nice DNA repair. If we inhibit PARP, a single strand DNA break will not be repaired, we'll get a collapsed replication fork, and the cell has got two options. Ideally it will undergo homologous recombination. However, cancers with a BRCA deficiency naturally cannot do this, so the only option is non-homologous end joining. This is error prone, leads to genetic instability to the point that the cancer just cannot survive. So we look at normal cells, the homologous recombination capable, and they are viable. BRCA deficient cells are homologous recombination deficient, but they have other repair mechanisms and they're viable. If we put a PARP inhibitor on normal cells, they are viable. If we put a PARP inhibitor on BRCA deficient cells, they die because they only have non-homologous end joining left, and therefore they die due to catastrophic DNA damage. So in summary, by knowing what is wrong with the tumour, we can get some clues as how we can pharmacologically treat the tumour. And we've seen that for homologous recombination and PARP inhibitors. We've seen that with APOBEC positive tumours, which might be more amenable to PD-L1. And there are some pharmacological chemotherapy regimes which are particularly suitable for mismatch repair positive tumours.